Hello everyone, my name is Seth Patterson, and today I'm going to be talking about DNA replication, transcription, and translation. So we're going to begin with DNA replication, which, as you can see, is defined as the process by which a double-stranded DNA molecule is copied to produce two identical DNA molecules. So these are just the general steps of DNA replication. We're going to go into much further detail in the following slides. We're going to begin with the breaking of the two DNA strands. This is the first major step for DNA replication, and it involves breaking the hydrogen bonds between the bases of the two antiparallel strands. This splitting happens in places in chains where are rich in A and T bonds. This is because there's only two hydrogen bonds between adenine and thymine, whereas there are three hydrogen bonds between cytosine and guanine. Having two hydrogen bonds makes helicase, an important enzyme that splits the two strands, job a little easier. Next, we're going to move on to binding of RNA primase. This is a very important step, the binding of RNA primase, and it involves RNA primase in the initiation point of the 3 to 5 parent chain. RNA primase can attract RNA nucleotides, which bind to the DNA nucleotides of the 3' to 5' strand due to the hydrogen bonds between the bases. RNA nucleotides are the primers for the binding of DNA nucleotides. Next, we're going to move on to elongation. And this process works two different ways depending on which strand you're on. For the 5 to 3' template, called the leading strand, DNA polymerase can read the template and continuously add nucleotides. For the 3 to 5' template, cannot read, DNA polymerase cannot read, so this is called the lagging strand. And in the lagging strand, RNA primase adds more RNA primers. DNA polymerase reads the template and lengthens the burst. The gap between the two RNA primers is called Okazaki fragments. Then we're going to move on to termination and repairs. The last step of DNA replication is the termination. In this process, this happens when the DNA polymerase reaches to an end of the strand. We can easily understand that in the last section of the lagging strand, where the RNA primer is removed, it is not possible for the DNA polymerase to seal the gap because there's no primer. So the end of the parental strand where the last primer binds isn't replicated. These ends of linear DNA consist of non-coding DNA that contains repeating sequences that are called telomeres. As a result, a part of the telomere is removed in every cycle of DNA replication. So that concludes DNA replication, and real quick we're going to move on to transcription, which, as you can see, is defined as the process by which the information in a strand of DNA is copied into a new molecule of messenger RNA, or mRNA. So these are an overview of transcription, and real quick we're going to go through each of these steps. So in initiation, RNA polymerase binds to the promoter sequence located near the beginning of a gene. Then a sigma factor is going to bind to the RNA polymerase, making it a hollow enzyme. This region of the DNA is unwound to form an open promoter complex. Here, phosphodiester bonds are formed between the first nucleotides, and there's no primer needed. This means we can move on to elongation, where the sigma factor separates from the RNA polymerase and becomes a core enzyme again. The core RNA polymerase loses affinity for the promoter and is able to move in the 3' to 5' direction on the template strand. And within this transcription bubble, NTPs are added to the 3' end of the mRNA. Lastly, we have termination. This is the end of transcription. We're talking about bacteria here, so prokaryotes. And in prokaryotes, there's two types of terminators. There's extrinsic, which is row dependent, and intrinsic, row independent. Basically, these are your ability to end transcription. Lastly, real quick, we're going to move on to translation, which, as you can see, is defined as the process by which a protein is synthesized from the information contained in a molecule of messenger RNA. So we're going to go over a brief overview of translation in the time we have left. Real quick, the ribosome binds to the mRNA at a specific area. Then the ribosome starts matching tRNA anticodon sequences to the mRNA codon sequences. Each time a new tRNA comes into the ribosome, the amino acid that was carrying gets added to the elongating polypeptide chain. The ribosome continues until it hits a stop sequence, then it releases the polypeptide and the mRNA. The polypeptide forms into its native shape and starts acting as a functional protein in the cell. Well, that's all the time we have today. Thank you for watching, and I hope you enjoyed this video.